My name is Elizabeth Vargis. I'm an assistant professor here at Utah State in biological engineering, and I do research in two major areas. One of them is optical spectroscopy, and the other is tissue modeling. So in the optical spectroscopy area, I use light to detect biomarkers of disease or to detect things like bacterial infection that are occurring in soils. In my tissue modeling area, I use different types of cells to see what happens with when the cells are stressed out in different environments or when malignant cells are grown in different types of substrates. So one of my students is a master's student, and he's working on developing Raman spectroscopy techniques to detect biomarkers of lymphoma and leukemia. So what he's trying to do is use gold nanoparticles to detect these biomarkers by enhancing their signal. And so we use just a laser and a really nice camera and excite those biomarkers with those little nanoparticles and enhance their signal.
Using Raman spectroscopy, you can detect certain cell surface proteins that will mark cancers. And so I'm interested in trying to detect these proteins so I can figure out what type of cancer I'm looking at. Because different types of leukemia and lymphoma have different cell surface markers, and in order to determine the subtype of cancer, you need to know the profile of those cell surface markers. There's methods such as immunohistochemistry, where you're actually placing a stain on the cancer cells, and you have to allow the stain to incubate, and it's expensive and time-consuming. Using Raman spectroscopy, you can just shine a laser at the cells that you're interested in, and based on the reflections of that laser, you can determine the identity of those cells. One of the downfalls of Raman spectroscopy is that it's very insensitive, so that the light coming off of the cells is not very, the intensity of the light is not very strong. So I'm using these gold nanoparticles. When you place those next to a cell, it will significantly enhance the light that's reflected off of that cell. So the eventual idea is to be able to get this to a point where it could be clinically implemented. So a patient would come in and you could draw blood from that patient and detect the cells within that blood to see if there's cancer cells. So it could allow earlier detection of blood cancers and also more specific detection and diagnosis.
Okay, so then I have a PhD student, and she's working on a similar technique to enhance Raman spectroscopy, so to enhance one of the optical spectroscopic techniques for bacterial detection.
The typical method to detect and identify bacteria is by plating on agar plates, and this takes anywhere from 1 to 2 days. And with laser spectroscopy, you can shorten that time down to 1 to 2 minutes. You can do that and also have an accurate idea of what you're looking at and also be able to treat it accurately.
She's looking at different types of bacteria that infect soils and how that can be translated to quick diagnostics in potentially non-laboratory settings. So if you're looking at contamination, or if you're looking at, potentially Department of Defense applications, if you're a soldier out in a specific area and you want to look and see what type of soil you might be interacting with, what type of contaminants you might be interacting with, this type of device could be used very quickly. Again, it's the same idea, being able to diagnose with high sensitivity really quickly instead of having to grow up these bacterial cells in the lab and then rely on some sort of pathologist or somebody that would visualize them. This will hopefully give people more of a quantitative way to see what types of bacterial infections are out there.
It's interesting because it's not necessarily a microscopic image; it's more of a spectra. And so your bacteria is giving off a number of different wavelengths, and the combination of wavelengths gives the bacteria a Raman signature, and this signature can be used to identify which bacteria you're working with. A lot of doctors prescribe antibiotics without knowing what they are trying to kill off. And so if they know what they're trying to kill off, they can prescribe the right antibiotic, and it can also help reduce the number of bacteria that are becoming antibiotic resistant.
Macular degeneration is one of the leading causes of eye problems in developed countries. One of the problems with it is that It's difficult to treat, number one, because it's the RPE, the retinal pigment epithelium, that causes dry, age-related macular degeneration, which is the more common type. And it's hard to get at it because it's inside the eye at the back, behind the retina. And the other problem is that it's also hard for drug treatments to get into the eye to treat the RPE because of the blood-vitreous barrier, which prevents big molecules from entering the eye. And it keeps your vision from getting clouded by like blood and everything leaking into your vitreous humor. But it also stops from a lot of medicine and treatments from getting into your eye as well. So as a result, not only is it hard to treat, but it's hard to even to test what treatments work. And as a result, there's no actual prescribed medication for treating dry AMD.
The plan that we came up with for his master's research is to study how retinal pigment epithelial cells are different between the eyes of birds and the eyes of humans. And so the cells are the same, but the way that they may be arranged is slightly different in the two different eyes. And so in avian eyes, the cells are kind of arranged in this tubular structure, and it kind of hangs out in the middle of the eye so there's no blind spot, and that is able to provide a lot more of the nutrients, the blood supply into the eye, and potentially avoid any of the diseases that humans get.
Birds have this thing in their eye called the pectinoculi. That's this, they have kind of, instead of retinal blood vessels, it's this thing that actually sticks out into the vitreous humor itself, like a big fan-shaped thing that is full of blood vessels that distributes oxygen and nutrients from the bloodstream into the retina there. And we were thinking maybe using that as sort of a microfluidic device to actually distribute, medicine to the RPE.
And so what we're just trying to do now is model both the human retina and the avian retina, and we're trying to use microfluidic devices. So microfluidic devices are going to be used for our research to simulate the blood vessels that are in the eye, and these retinal pigment epithelial cells are what kind of line one of the sides of the blood vessels. And so we're using it in two different schemes. So one is just kind of a normal blood vessels, kind of like what you envision, kind of this branching pattern, and that's to simulate the human retina. And then one is the more like a fancy tube-like structure that is in the avian eye. And so we're just trying to use both of those and see how the eyes, or how those cells react.
What we want to do is come up with sort of an artificial RPE, our retinal pigment epithelium that we grow in the lab that we can get at, without having to cut somebody's eyeball open. And so that we can test different, so that different drug treatments can be treated on RPE cells and their effects,
What I want to do is understand disease and then try to detect it with that understanding. And so even though the two areas seem kind of disjointed, so that there's the tissue modeling and then there's the optical spectroscopy, the tissue modeling is going to help me understand what causes this disease and what are the markers that can be used to diagnose the disease. And then the optical spectroscopy can be used to actually diagnose the disease. And so I'm looking for a way to combine the two, and I think that'll just take a motivated PhD student or even a couple of motivated undergraduate students. But that's what I'm looking at, is to really develop tools to be able to diagnose and detect either disease or pathogens or even bacteria that we want to find in certain areas. So these contaminants in soil or in food or in the air or in humans. And in the tissue modeling is just to understand what causes disease, what we can do to prevent or treat it.